So hi everyone, thank you very much for coming to this presentation. It's really great to see your interest in project discovery, both here and both yesterday, where we showcased the client, the minigame. And we're super excited to be here, as this is actually the first time that we can show you what we were working on. With this, I think we are at the most important milestone of this project to bring real-life science into EVE Online. This March, in Reykjavik at FemFest, we presented an idea and a working prototype to show you how we can imagine integrating scientific problems into the EVE universe. And now we are here after half a year, and we can show it actually working in the EVE client, and that is just awesome. So back in Reykjavik, I finished my presentation with this slide, which was my call to action slide. And I told everyone that this is going to be a reality only if you guys support it. And now we're here, and I'm happy to say that CCP saw this support and decided to give a go to the project. And this is just incredible for us. The support and enthusiasm that surrounds this project, both from the player community, from CCP team, and from the researchers is way beyond our wildest dreams. So thank you for that, and I really hope that we can live up to the expectations. So yesterday before the keynote I saw, actually it was 2 days before, the keynote we gave the Hands Up, who was in Reykjavik. And I saw that many of you couldn't come, so I thought to make a small summary of what the project is about. So first I have to talk about citizen science. Citizen science is when us, outsiders, help scientific research by doing something. And this something can be classifying images, or transcribing handwritten text, or taking photos of animals. Nobody needs really a formal education in the field, so we can just use our cognitive skills, our brain power, but it really boosts scientific research. Small scientific research teams simply don't have the manpower to deal with the amount of data. But luckily, there are the citizens who are there to rescue. Here I just picked some random examples of what kind of projects are going on in citizen science. There's Galaxy Zoo, which was among the first, and it was a project launched to classify galaxies. It became so successful that later it became the foundation of the Zooniverse platform, which now hosts tens of different research topics, for example, the snapshot Serengeti. And then there is another very well-known and very successful project, the Foldit project, the protein folding game, where we help researchers to understand the three-dimensional structure of proteins. Now, why is citizen science important? Obviously, there are the results that we give scientists, and that is a very valuable input. But you can say that So you can say that, and actually it came up as a topic that with the pace that machine learning is advancing today, this will be kind of a useless activity relatively soon. And I want to go back to this image that I used as a background just before. This is a beautiful image and a very interesting one as well. And if you magnify it, you see that these are galaxies and astronomical features. And in 2004, a paper was published about an algorithm called BRUT, whose aim was to identify bubbles in space. And the interesting thing is that the project used 35,000 citizen scientists' input to feed the machine learning algorithm. So actually, in the long run, citizen science is very important to support machine learning, because we can generate large and good quality data sets. Another specialty of citizen science is or I would say the human cognitive skills as compared to algorithms, is serendipitous discovery. That's when we discover something which we're not looking for originally. It happened in science many, many times. For example, penicillin is something that we can thank to these lucky accidents. Or something more close to Eve is Henny's warburg, which is a really, really rare astronomical feature. And one citizen scientist found it while classifying galaxies. As great citizen science as it is, there are huge challenges. And the two biggest challenges is how can you get users to help your research project? How can you get people on board? And once they are there, how you keep them engaged in the long term. And unfortunately, citizen scientists tend to disappear from this project very, very quickly. And that is a huge challenge. And to answer these challenges, we came up with an idea. And our idea was to take these scientific problems and inject them into major computer games as a seamless gaming experience, integrating them into the visuals, with the narrative, with lore, with the game mechanics, and of course with the reward system. And you know, games are huge. I mean, we people play billions of hours every week. And we know that if we manage to transform a small percentage of that time to something useful in science, that's going to be huge. That's going to be the biggest human computation engine supporting science. So we came up with this idea. We created MMOS, which is a small company, which stands for Massively Multiplayer Online Science. And we started to work together with CCP on this project. So Project Discovery. For Project Discovery, we teamed up with the Swedish research group, the Human Protein Atlas. They have millions and millions of images, microscopic images. And we chose the subcellular atlas because they had these beautiful, mind-blowingly beautiful images. They were a natural fit with Eve. We didn't even have to transform the images. Also, we picked a relatively simple problem because we didn't want to go into implementational traps. We didn't want to fall into implementation traps. Rather, we wanted to test whether this concept of putting scientific problems into games work. We have about 250,000 images just in this database. And the task is to classify these images. There are 27 locations where proteins are attached to the cell. And that's what we have to find. Actually, the science lead, Emma, is here to talk about the science behind, so I don't want to talk about it more. I can't even. But what I learned about this project is that one scientist told me that in the last 20 years, the biggest effort in biology was to map the human genome. And now the focus shifted a bit to proteomics, because actually it's the working molecules, the proteins, who can make the nasty stuff if there is a problem with the DNA. So Professor Landberg is here, and that's her. Actually, she's working for the Human Protein Atlas, but in the meantime, she'll have a permanent position at the Sisters of Eve. And she'll talk about the science. There is one thing I want to tell about this that she probably wouldn't talk about, is that she actually canceled a conference in proteomics to be able to come here and talk about the project. And for me, it shows more than anything that how important this is for researchers. What is also very interesting about this project is that we have these 250,000 images, and a relatively large part of it is already analyzed by experts. So we have the gold standard. Then we'll have players' opinion from this project. I was talking to Chris Lintott, who is the founder of Zooniverse, and he's open to run the same project on Zooniverse. So this is not just going to help to build a human protein atlas, but I think this is going to be the biggest research material in citizen science itself. We can learn a lot on this. Uh, I wanted to talk a bit about the project structure, which was a very successful cooperation, not only between CCP, Human Protein Atlas, and us, but from the beginning, CCP Seagull suggested us to team up with the Reykjavik University. So actually, the in-game implementation of the minigame was done by students from the Reykjavik University. And it was such an appealing project for students that we were really happy and lucky to work with the best guys in the year. Unfortunately, they cannot be here. They couldn't come to Las Vegas, but if you can make it to Reykjavik next year, I really hope that you can meet them in person, because they're great guys. Also, since this is a tech-savvy audience, I thought to talk a bit about the technological background. We created an API for the project, which does the heavy lifting of the citizen science side of it, like allocating the tasks, gathering the results and aggregating them, and also to keep track of player performance, which is very important. On the other side, there is the Eve components. Our API mostly talks to the Eve server, so we are kind of protected. And then the game client only fetches the images directly from our image database. So, but this is an experimental project still. We're very in the beginning, so please be gentle with it. Don't try to hack it the first day it comes out. And one more thing is very interesting with this setup, that we realized that by having an API for citizen science, we can target other audiences. For example, we started to work together with MOOCs, which are these massive online courses, where we inject real-life science in the assignment section. Again, very, very, very interesting. In the meantime, I know that Eve is not only about being nice and gentle and altruistic. So it was, from the beginning, one of the biggest questions, how we protect the system from being exploited, for being it's used only to get Sisters of Eve loyalty points. And this question was raised already in Reykjavik and also in the Reddit thread that was started just after Andy's keynote speech. So we worked on that a lot. We have some solutions. This keeping track of the user accuracy is part of it. I don't have the time to go into the details here, but we'll have a roundtable talk just after this. So please come if you're interested. We're really happy to answer these questions. Peter and Emma has a lot to say, so I'm soon finish this. I want to finish it with a really lousy player word. At Reykjavik, Hilmar was announcing the motto of this year, less hype, more substance, which I really think that CCP made up to this promise. So my version is less hype, more science, and really hope that we can do that this year. And without repeating my first slide, my call to action slide, the message remains the same. So we want this project to be successful, simply because we think it's very, very important for science. So if we are succeeding, that's going to change a lot of things in science. If we fail, then I think no other gaming company will risk to give it another try in the following years. So to do that, to succeed, we need your input. On one part, we want to make a good game. We want you to enjoy this part of the game, because we think that is what makes it engaging in the long term. And also on the other side, we need to provide valid data for scientific research groups. So we need input from you to how we can help to give more training materials, background info, to help you to make the classifications right. And I think if we manage to find a solution, then we can create the biggest human computation engine for scientific research, and in the meantime, we can ship you cool in-game features. Thank you.
Can I maybe just use this mic? Yeah. This works? Hello. Hello. So, I'm CCP Scarpia, lead game designer for EVE Online. Gives me a great pleasure to talk about this project with you all. I don't know, has anyone tried it? Raise your hand if you tried it, or if you're here at FanFest and have heard about this before. Okay, cool. So I'm just going to recap a little bit, like Attila, some things we talked about at FanFest. But I'm actually really looking forward to hearing from Emma and actually getting more, for us to be able to actually share with you more about like what this research project actually does in the real world. Because that's what it's all about, and that's what sort of is our main incentive for actually doing it, and hopefully you actually taking part in it. So, brief overview over the project, what's behind it, and the sort of step-by-step walkthrough of our EVE Vegas demo. So when MMOS approached us about this awesome idea, I don't think they expected it. I think they might have expected it to take us more convincing. But to us, this was really a perfect fit for EVE, and we had to give this unique opportunity a real shot. But how would we actually build this? Like, while we believed in the idea, there were still a lot of unknowns, and it's not just like any other game feature in EVE, so actually pulling some people off of EVE development to work on this seemed like a really risky proposition, and yeah, it was really risky. So, like he said before, we were really fortunate that we had students at Reykjavik University who jumped on it, took it upon themselves to make a prototype in collaboration with us, and that's what they showed at FanFest. And then after FanFest, they actually went on to implement it inside the client. And then in the fall, we had 2 new students who took over and are now working on it. And we're like together marching towards a CC release as soon as possible. And a polished TQ release sometime in the winter. But of course, as we were starting out, one of the first things we had to identify and decide on was the criteria for what type of research project would qualify for the citizen science in EVE. So the things we sort of identified was, first, it had to be something that was charitable and altruistic in nature. We didn't want to risk doing anything that was sort of like self-interest, or we just wanted to be altruistic or in the name of like pure science. We wanted to make sure that everyone could get behind it, and there were no, and that we knew what was behind it. Second, the images and other research data had to be aesthetically fitting for EVE and like easily adaptable thematically to the universe. So having data that we can easily manipulate or that just fits right into the world is much better than something like this, which I would love to help out with, but we couldn't, like, we can't use like actual like wildlife photos or a number of other things that we actually considered. Third, we felt like the task to analyze the images or the data had to require like a reasonably consistent length of time, at least within a project, so that you as a player can actually build up an expectation of how long it takes you to participate in it. I remember I did a bunch of citizen science like many years ago, like with Galaxy Zoo and some others, and I remember Some of them, yeah, like LSU2, once you get really good at it, it takes you like 5 to 15 seconds to do each one. But then there are some that took like, sometimes they took 10 seconds, and if you want to like do a good job, they sometimes took like 10 minutes or 15 minutes. And that would hurt the gameplay a lot, I think. Right. And then finally, while we would like to be able to actually do more than one project and actually swap them out once, if a project actually finishes, we felt like it was much better suited to have a project that has like a huge volume of tasks, like the one we picked, which is, as like you said, we have like 250,000 images to actually analyze. So the third partner here is like Attila said earlier, is the Human Protein Atlas. And yeah, I also can't and won't go into it right now, but I really want to show you images here. Like as soon as we saw these, we were like, yeah, this is perfect. Our art director was like, he thought that we doctored them, or like somebody had made some magic, but he was like, wow, this is awesome. You don't have to do anything. These are basically Jovian tissue samples. Literally, that's what they're called in the game. So yeah, this project just has this endless source of unique raw images that are just completely unpredictable, and they're really, really beautiful. So let me just show you a quick run through what it actually looks like in the client if you haven't tried the demo out. I think the demo is still running in the next room. If it isn't, then I'll make sure to put it up right after this, so you can try it out. So first, you just initiate project discovery from the Neocom. You get welcomed by Sisters of Eve. Of course, I don't know if I have to qualify with it. This is all still, there's lots of work left to do before CC. But yeah, it's looking it's getting there. It's actually getting pretty close. So yeah, you get welcomed by Sisters of Eve. The head of the project is Professor Lundberg. She will be the one who will communicate with you and teach you the game and hand out rewards. On the left, you have the image that you're supposed to analyze. Beneath the image, you can actually filter the different color channels to help you with the analysis. So you can only see like the green channel or the green and the blue, etc. On the right, there are the four main categories, and each category has a number of different samples. I think the total is 25, right? Yes? Yes. Okay. And you have to select like one or more of these before you actually submit. And then you can see that only a few of them are actually lit up right now. It's because you're in the tutorial phase. And in the tutorial phase, you only get like a handful of them. And these are all like we know the actual result for these. So while you're going through the tutorial, we just want to teach you a few at a time. So as you can see, the tooltips, there's a tooltip for each sample. It tells you what you should be looking for, like what characteristics you're looking for. When we go on CC, we're working on actually expanding those tooltips to have these annotated versions. So it actually tells you a little bit, actually a lot better what you're looking for. And I felt personally it was much clearer for me to actually, and I learned a lot more when I actually had these. This is the cheat sheet that we made for eVegas. You can also see it over by the demo. So yeah, work in progress. As you progress, you level up in ranks. And on the top right, you can see the accuracy meter. That's actually, this accuracy can go up and down, and it reflects how well you compare to others. Yeah, just how accurate you are, basically. And that's really the stat that you're going to care about, because that's going to that scales the rewards that you get. both to, of course, reward people who do really, really well, but also to make sure that people are not just haphazardly clicking through it and trying to do it really quickly, because as soon as you drop below a certain threshold, you no longer get any rewards until you build it up again. So you need to do well. There's no incentive in just rushing through it. So as you make your selection, you sometimes see other sample images actually get crossed out. This means that they are mutually exclusive. So now, like I made 2 selections, and it means that when I picked one of them, another one got crossed out. Then you submit it, wait a few seconds, and you get the result screen back. Now you get this result screen because these are solved images, so we know the answer immediately, and we can tell you that, like, I made one correct one, I made one incorrect, and then missed one. But of course, yeah, and then you get a new image. Yeah, but of course, yeah, when you graduate from the tutorial, you start getting images that haven't been solved yet. And we won't know maybe for a day or 2 or 3, like what your actual score is compared to everyone else. So to replace the feedback that you get after you submit it, we instead then just show you how you compare to everyone else who has actually submitted a result up until this point. So it's not until like an amount of analysis has been made for each image that a consensus has actually been reached. So you can see here, like I made a lot. I made like 6 different selections, and they pair up like with like 100% of everyone who's done this image, pick the one in the center, and etc. You can see sort of the spread. Yes. But I don't want to take up more time. Emma is here, and she's going to tell us why we should actually. She's going to show you why this actually hit home for us, this specific project, and why we should participate. Emma. There you go. Thank you.
Thank you. It's great to be here, I have to say. I've had a lot of fun, and I've learned a lot by showing the demo to you and talking to you, so thanks for having me here. So I'm going to talk about the science behind the project. And first, I'll just introduce myself. So I'm assistant professor in cell biology proteomics at the Technical University in Stockholm, the Royal Institute of Technology. And I work at Science for Life Laboratory, a big research center focused on life science research, high-throughput life science research. And my main work is being done within the frame of the Human Protein Atlas project, where I run one of the 4 sub-atlases, the subcellular protein atlas that you've seen the images from. So to start out with some basics, you probably all know that our genome was sequenced, and it's almost 15 years ago now, so in 2001. And the current estimate is that we have about just below 20,000 genes, and each gene code for 1 or more proteins. And we are interested in the proteins, because that's what we see in the images, and I will explain that to you later. So why do we want to study proteins? Because, as I think Attila said, that the proteins are the actual doers. They do most of the things in our body. They can provide structure, like in hair and muscles. They do signaling, insulin, growth hormones. We have enzymes. Our immune system is based on a lot of different proteins. They perform the regulation of the DNA and the gene expression and everything. So they're the doers, and that's why we want to study them. And also, protein malfunction or malstructure, any problems with proteins often end up with some kind of disease. And most drugs on the market today, they act by targeting a protein. So there's if we would be able to characterize all human proteins, that would help us a lot to understand the human body and understand how it works, and also be able to understand why disease develop and hopefully design better drugs. And well, that's a large perspective of why we want to study proteins. And you might think that 15 years ago, if the genes were known 15 years ago, don't we know the proteins by now? And actually we don't. 30% of all genes, for 30% of all genes, no one has even detected the protein in any assay, any sample, any condition. So there's quite a lot of work to be done there. And one of the reasons that the proteins haven't been studied as much as the DNA is that it's a more complex task for many reasons. But one of the reasons is that the genetic content is more or less the same in all our cells. That's why you can take a blood sample or a cheek smear and analyze your DNA. But it's the expression of the genes, so the protein content that makes up the differences between the cells. So different proteins will be present in a brain cell than a skin cell. So if we want to study the entire human proteome, that means that we have to have samples that covers the entire human, more or less, and then it becomes very complex. So, but luckily enough, nature is often very clever. So humans and all multicellular organisms has been built up by compartmentalization. So we have organs like the liver and the heart, and they perform specific functions, and they are in turn built up by tissues with specialized functions, and they are in turn built up by cells with specialized functions. And the cells have different organelles that all have specialized functions. So if we could make a map and see where the proteins are in our body, we could get clues about the function. So if I would find out that a protein is in the mitochondria in the liver, then that I can start to make some conclusions about the function of that protein, or maybe we can at least start to try to understand the proteins and the complexity of the human proteome. And the field that we're doing research in is called spatial proteomics. So it's all about creating those maps, and we want to answer questions like, where is the protein expressed? Is the distribution static or dynamic? And how do the distribution of this protein relate to all other proteins? Because if two proteins show a very similar pattern, it means that they might actually be involved in the same biological processes, and they might even interact. So that's what we're interested in. So it all comes down to pattern recognition. And the Human Protein Atlas project, it's a Swedish project, so I work in Stockholm, and it's headed by Professor Mattias Ullén. It was started in 2003. It's funded by a non-profit organization, and the Wallenberg Foundation. So what we do, basically, is that we generate probes called antibodies, and they have the capability of specifically binding to a target protein. So we develop one probe per protein, and then we can use those probes in different assays to generate these maps and these images showing us where the proteins are. And this is the Protein Atlas team. We're about 100 people. It's a great project to work in. And it's been running for over 10 years now, so altogether we spent more than 1,000 years in this project. So it's not a small, it's a very big research project. So we need your help to get as much data as possible out of this project. And I'm not going to go into the details of what we do or how we do it or anything. It's very multidisciplinary, and I think the common denominator is that we're always at the forefront when it comes to automation and state-of-the-art technology. So everyone that is working in the project is quite interested in pipetting robotics and things like that. But in the end, we use the reagents, the probes, the antibodies that we developed in different assays, IHC, immunohistochemistry, and IF, immunofluorescence, that's the images that you've seen, to map out the proteins. So I will show you some examples. This is an example of tissue. This is fallopian tube. And you can see we have targeted a protein here, CD47, with an antibody, and the antibody gives a brown color. So here you can see that this protein is expressed at the surface of the fallopian tube, and you get a single cell resolution. So you can see that this protein is expressed not in every cell, but maybe in every other and every third cell. So we get a lot of biological information from looking at this image. But it's very rich in information also, and it's complex to extract all of this information. So we need your help. This is an immunofluorescent image. So this is a human cell line. And in blue, you see the DNA. So here you see the chromosomes. And in red, you see the microtubules. this cell is about to divide. So you can see that the red part is about to pull the chromosomes apart, and this cell will divide into two. And then the protein we're looking at here in green is called BUB1B. We can see that it's localized actually to the spots where the red attached to the chromosomes. So here we also get a lot of information by just looking at the pattern of the protein. And these are the types of images that you're supposed to help us analyze in Project Discovery. So altogether, we have 13 million images in the Protein Atlas in the database. And you can find the database at proteinatlas.org. So it's a publicly available database, free of charge. So what we do is, in this project is that we make these maps, and we put all the data in our database, and we hope that other researchers will use this database when they study cell biology and disease and, well, all life science research, more or less. But we are mainly focusing on creating this catalog. So it's one of the larger biological databases. We have almost 100,000 different users every month. So pay it a visit if you haven't been there, and I'd be happy to show it to you if you want me to, so just let me know. And actually, so when I discussed with Attila in the beginning, we decided that we need a very confined problem for the Project Discovery. So we decided to not work with the tissue images or the cancer images right now. So there's 4 different atlases. To the left, you have the tissue atlas, the subcellular atlas, a cell line atlas, and then a cancer atlas. So we decided to work with the subcellular atlas because it's simpler. You have single cells at least. You don't have a mixture of cell types. And if it works out well, we can proceed into the other types of images. And again, it's a lot of, we've spent the last 10 years just optimizing procedures and what we do. So we cultivate human cell lines. We stain them with these probes. We take high-resolution confocal images. And now our major bottleneck is to actually annotate and interpret the patterns in these images. And I will show you a lot of images now, just to kind of give you a sense of the patterns that we're looking at. So this is, we use some reference markers just to outline the cell. So we always stain the nucleus with blue. So here you can see 1 nuclei, and it's about 10 micrometers in diameter. And then we stain the microtubules in red, part of the cytoskeleton, and it outlines the rest of the cell. And then we actually have one more marker that is not in Project Discovery, so you can't see this channel in the minigame. But it's the endoplasmic reticulum. So on top of these markers, we stain the human proteins one by one, and it might look like this. We always show it in green. So this is, when I look at this image, my conclusion would be, oh, this protein is localized to the cell membrane. And similarly, I would look at this image and say, oh, a cytoplasmic protein. And this is a mitochondrial protein. The mitochondria produces the energy for the cells, so maybe this protein is involved in energy metabolism somehow. This protein is localized to the endoplasmic reticulum. And the Golgi apparatus, the sorting machinery of the cell. We see vesicles. We don't know what type of vesicles, but it contains this protein at least, and it's probably going somewhere. We can see the different cytoskeletal structures. This is the microtubules that, for instance, cytostatic drugs, they act by disrupting the microtubules. So the microtubules are very important for cell division. We have other types of filaments. We have actin filaments that are important for movement cells. And we have intermediate filaments that provide stability for the cells. We can see centrosomes that are the center of the microtubules, very important for cell division. You can see the cell surface, plasma membrane, important for interacting with other cells. It's the surface of the cells that first receive all other signals from the outside, so very interesting from a pharmaceutical point of view. We have proteins at the focal adhesion site, in cell-cell junctions. A lot of proteins are in the nucleus, about 1/3 of all proteins. You can see proteins in the nucleoli, subcompartment of the nucleus, or in the nuclear membrane, and so on. So these are the types of images that will be in Project Discovery. And what we want you to help us to do is to recognize this pattern and kind of map them onto a canonical cell. So we would want to, well, make a model of a canonical human cell where we could see how and where all proteins are expressed. And often, the images that I just showed you, that's pure patterns. There's one pattern in one image, so they are very easy to classify, but often it's way more complex. So we really need your help. And just to give examples of how this information can be used. So the output for us is to show the images in the protein atlas and also show the locations that we have classified. But other people then use this information. For instance, mitochondria is a very important organelle for heart failure. So mitochondria produces energy, and our hearts need a lot of energy to work. I think that in average, a heart uses 30 kilos of our energy molecule ATP every day. So your mitochondria in your heart needs to produce 30 kilos of this molecule every day. So if the mitochondria in your heart doesn't work properly, you might have heart failure. So we're collaborating with researchers that are studying heart failure, and they are of course very interested to know which proteins can we find in mitochondria in heart, because then they can make targeted studies of those proteins to try to understand heart failure and develop better drugs. Same thing, for instance, for proteins localized to the centrosomes, the center of the microtubules. They are very important for balanced cell division. So there's a group of diseases called celiopathies that can end up with like extra digit or even more, way more severe diseases. And they are formed when you have a malfunction of a centrosome protein. So here we also collaborate with researchers to try to find all the centrosome proteins, especially the ones that localize asymmetrically. And now we're down to very fine level of annotation and pattern recognition in the images. So these are the types of fine details where we want you to help us in project discovery. So in the end, we generate a lot of images like this every day, confocal microscope images. And as you can see, all of them looks different. They're very rich in information, and there's a lot of variation in the images. Even though these are cell clones, there's still variation in the images, and we need to capture that information. So what we ask you to do, as they explained already in project discovery, is you get a reference, you get one image, and you're supposed to click on the reference structures in the cell that you can find in that image. And you have 25 different structures to choose from right now. So this is more than we have in the protein atlas. Right now we have 18 different structures annotated, so we're expanding to 25 here. And so the aim of project discovery is both to identify more detailed patterns and to identify cell-to-cell variations, and also to compare the performance of our annotations. We're a few experts, with 2 people looking at each image, and then compare it with computers. We do some computational image analysis, machine learning approaches, and also compare it with many gamers to see how we perform against each other. So it's a big citizen science experiment. It's very fun. So I think one of the areas where we will gain a lot of information from your help is when we have mixed patterns. This is hard to analyze with computational image analysis. Here we see the nucleus, the Golgi apparatus, the cytoplasm, and the plasma membrane. So by having a lot of people looking at very complex mixed patterns like this, I think we can find many more details than we do by only having 2 experts looking at them. And this is something that we have found in the protein atlas, that the number of proteins with mixed patterns, it's much higher than we expected in the beginning. So it seems to be a very complicated balance of how proteins are distributed in the cell. So it's fun from a systems biology point of view to mine this data. We also ask you to refine the nuclear patterns. Right now, we only classify nuclear proteins into nucleus, and that's quite big. It's 50% of the volume of a cell, so of course we want to refine that. There are a lot of different patterns. You have distinct spots that might be DNA repair proteins, and you have speckled patterns and different nuclear bodies and everything. So this is something that you will help us to do, because we don't have the time to reanalyze all images for this purpose. This is examples of different cytoskeletal proteins, and as you can see, the images look very different, the patterns look different. So here is also, for the microtubules, you will help us to subclassify the microtubule patterns, because this we don't have the time to do ourselves either. So it will be subclassified into many different smaller structures. And sometimes we still find uncharacterized structures that we don't know what it is. Now we know what it is, but we didn't in the beginning. So at some point, we might also add kind of, I found something new, and that you can mark it in the image or something, so that you can help us to actually discover new things that we didn't know of. And one more thing that you will do that we haven't done ourselves already is to identify proteins that show cell-to-cell variation. And the most likely reason for this variation is that the proteins only are expressed during a certain period of the cell cycle, the cell division cycle. Because these are static images, but they're not synchronized, the cells, so they're in different positions in this cycle. And patterns typically look like this. So these proteins are very interesting, of course, because, well, cancer is a disease, proliferative disease, where cells start to grow without control. And of course, it's interesting to identify all the proteins that seems to be expressed in correlation to the cell cycle and distinct phases, because then we can understand the cell division process better. And it can also help us to understand cell biology a lot better. So we'll also ask you to identify when you see patterns like this. So in the end, what we want to do with this project is to refine the details in the protein atlas, identify cell cycle dependent patterns of protein expression, and compare the performance of you, us, and computers. And in the end, we hope that the data, if you perform well, your annotations will end up in the protein atlas. I think it would be great if you could look at an image in the protein atlas and you could also see that the gamers classified this, when you look at one protein, it could say that gamers classified this protein to have the most similar pattern to this protein. Kind of like when you're on eBay and you can see what other people bought. That would be very nice. And I think that's something that researchers would like to. So, and if it works well, we'll start out with the subcellular images. If it works well, we can expand into the way more complicated tissue images and also the cancer images in the cancer atlas, because they are completely unexplored today. So I would like to show the cell profiling group. This is the group of people that generates these images and that annotates them, and that will help to answer questions from you and so on. And also thank the funders, Knut and Alice Wallenberg Foundation, for funding this project. And also thanks to all of you for being here.